And welcome to IntoBio lecture exam for review sheet. So this is a review sheet for the final exam next week. Okay, so for each of these, and this comes from the review sheet that's posted on Moodle, I'm going to try to emphasize what you should know and the types of questions. So it's going to be 50 multiple choice, just like the other exams. There will be a restriction endonuclease problem, which I'll get into in a second. The first thing that I would ask, and a couple of multiple choice questions, would be negative feedback in the control of blood sugar levels in humans. We covered it in lab, and I had mentioned that it's going to be a couple of questions on this exam. Negative feedback meaning that if you have a uptick in blood glucose, then that's going to start a feedback loop that will lower that glucose level in the blood back to normal homeostasis. If the blood sugar level drops, then the response for negative feedback is the opposite, so that what will be set in motion will be a rise in blood glucose to get it back to normal. So if blood glucose is high, then the pancreas secretes insulin, which binds to liver cells. And remember that liver cells or hepatocytes are able to take excess glucose from the blood and then to bring it in and polymerize it into a glycogen chain for storage, like beads on a necklace. And so the insulin binds to the liver cell and causes the polymerization of the excess glucose that it gets from the blood and makes a glycogen chain for storage. Now, if the blood glucose levels suddenly drop or low, then the pancreas secretes a different hormone, glucagon. Again, it can bind to the liver cell, and what it does is depolymerize. In other words, you have too little blood glucose. It's going to take the glucose stored in the glycogen chain, break the chains, release the free glucose to the blood, and then the blood goes back to homeostatic normal. So the glycogen chain then is broken, frees the glucose, returns it to the bloodstream in order to reestablish a homeostatic normal levels of blood glucose. Okay, so a couple of multiple choice. Epigenetics, that could be both, and I'll get into the matching part in a second, probably 5 or 6 multiple choice questions on this. So we know that epigenetics means that you can change things above epi, the nucleotides, the A's, T's, G's, and C's of the DNA double helix. So it is heritable. In other words, parents can pass on epigenetic changes to their children since it's in the sperm or egg. It's non-genetic, meaning that it's not going to alter the A's, the T's, the G's, the C's information, but modify the histones and modify certain sequences called CPG islands, but they don't actually hit or mutate the nucleotides of the DNA. So epigenetic changes can persist and be passed on, and you can either methylate the DNA, which can also be called chromatin, and by certain mechanisms of changing and tagging, not changing the DNA, but tagging the DNA or tagging the histones that make up the chromatin complex, you can either turn on or turn off gene sequences on the DNA. So gene transcription can be the result of different histone modifications. So the thing is that you can either inhibit or you can activate, and the thing is that normally, let's say for my red blood cells, I would want the genes that code for hemoglobin to be activated, but the genes that coded for neurotransmitters to be inhibited. All right, so let's get to the next slide, which goes into some specific mechanisms. Okay, I need you to know this. So the modification of cysteine, or cytosine, sorry about that, by adding a methyl group, that's methylation, will inactivate the gene. And if you remove that methyl group, these are the CPG islands that they're talking about, that will reactivate the gene. So that's the other thing too. So I'm studying for this exam, I'd study your notes, I'd study the review sheet, and then listen to me blurb. Okay, so histone modification, in other words, if I acetylate, put an acetyl group on to certain histones, then that will activate the gene. So these are the two that I want you to know. Inactivation by methylation of histone, or acetylation of the histones activating. Okay, so everybody gets an imprint of their epigenetic modifications. And so you can silence or activate a number of genes. So the CPG methylation, the histone modification by acetylation are ways of imprinting genes. And let's go back. And aberrant imprinting can cause abnormal development and disease later on. And as well as parental passing on of epigenetic traits, the person during their lifetime can also pass on epigenetic traits. And the environment can also cause changes that can affect a person in their lifetime. Okay, so what are some applications that we have found now that we understand better the mechanism? Epigenetics in cancer, neurological disorders, autoimmune disorders. Those are the big three that are actively being researched. So with epigenetics in cancer, I want to emphasize, yes, cancer can be caused by oncogenes, can be caused by inactivation of tumor suppressor genes. These are mutations of the A's, the T's, the G's, and the C's. But in addition, we now realize that you could have just perfectly fine DNA sequences, no mutations whatsoever, and the cancer is caused by epigenetics, either inappropriately activating or inappropriately silencing genes during the person's lifetime. So cancer, right? Mutations in the DNA, that's what we think about, but now we realize it's more complicated. So the changes in the DNA could induce genomic instability, and so this is an example of an oncogene that should not be expressed, but hypomethylation, in other words, they should not be expressed, but hypomethylation can cause unwanted expression of the oncogenes. Moving right along, neurological disorders. And this can be in the realm of psychology as well as biology. So abnormal epigenetic processes are linked to neurological disorders such as neurodevelopment or neurodegenerative diseases. Neurodevelopment could be fetal development. A neurodegenerative disease could be Alzheimer's. Several neurodevelopment disorders are the result of dysfunction of specific epigenetic modifications and can lead to disorders. Now, this could be, again, behavioral disorders, psychological disorders, schizophrenia has been studied, as well as others where it seems that certain neurotransmitters like serotonin are underexpressed, and epigenetics is being looked into that because we actually don't have drugs yet that are able to address epigenetic changes, and we need them. Okay, so autoimmune disorders, that would be from rheumatoid arthritis to lupus to diabetes type 1. So this is when the immune system turns on you and starts attacking certain parts of your body. And epigenetic aberrations now have been discovered. It's almost as though we needed to know that it existed. Epigenetics is a real thing. And then we were able to fish for those disorders. Because every disorder, let's say, we'll take Alzheimer's, there are always subgroups, there's always subcategories. And one of those subcategories is in the realm of epigenetics. So hypomethylation sometimes can turn on a gene in an aberrant way, and so forth and so on. The thing about all of these three things is that they're new sciences. There's a new way of looking at diseases in certain populations, not in others. But the more that we have tests, the more that we have drugs, the more that we're going to be able to modify that epigenetics, and it also could be therapy. This is the thing. We're really at the very beginning of this. So epigenetic treatments, we do have some, they're just not, they're either in development or they're in teaching hospitals and they're relatively new. Hopefully in your generation, they'll be every day. So you have tailor-made therapy strategies, and the whole idea is to try to take a disease that has a modification that's epigenetic and to reverse it. So some of these, like the cancer treatment, there are some trial chemotherapy out there for people who test positive for epigenetic cancers that can inhibit methylation or inhibit acetylation. But in other words, if you can start to modify very specifically where you get the histones and epigenetics and acetylation, and you can either up or down regulate it, then you'd be able to treat the patient effectively. All right, we're on to the next one, which is recombinant DNA technology. And here we know that in the lab that we did, we took a piglo gene from a jellyfish, and it was inserted into a plasmid that is bacterial in origin, as well as an antibiotic-resistant gene for ampicillin. And you were able to heat shock it into E. coli, and those E. coli's that then were able to seal their membranes and grow and divide were able to express ampicillin resistance and to express the piglo under UV light. And you did a fantastic job. You got recombinant colonies, they glowed in the dark. And so now, with that backdrop, let's talk about the DNA technology. Some of it you did, some of it you'll do later in genetics. So the restriction endonucleases are bacterial in origin. They developed this, bacteria developed this in order to help destroy foreign DNA like viruses. So we, however, have learned to use these restriction endonucleases to cut genes from humans, and the human DNA can be inserted into bacterial plasmids. It has revolutionized therapeutics in medicine. So what is a restriction endonuclease? Well, it's an enzyme that cuts DNA in a particular sequence at a particular site, and it's made, obviously by bacteria, and now we can separate them from the bacteria and use them in the lab. So, for example, and I would tell you this on the exam, BAM-H1 recognizes the DNA sequence GAA-TCC and cuts after the first G from 5 to 3 prime. All right, then what the bacteria did is that we were able to insert that plasmid, and when it was inserted, what you will do when you take genetics in a couple of years, you will seal that inserted gene, whether it's a piglo gene or a human gene, with ligase. And then you can pop it back into the bacteria. All right, so some background on restriction endonucleases. And this section is mainly going to be multiple choice. And the restriction endonuclease problem, there might be a couple of them, will be on the next slide. So we know that it cuts DNA into pieces, it has a specific recognition site, and then once it inserts the gene that it wants inside that plasmid, it'll seal it up with a ligase. So we know that the sticky ends. We did this in lab. You saw the sticky ends when you put the therapeutic gene on paper into the E. coli plasmid. You got them to pair with each other, and then in real life, they would be brought together permanently with the ligase. Host vector, that means let's say human insulin. The host is going to be a bacteria, and the vector is going to be how you change the host. Now, the vector we used was a plasmid. So we use E. coli, we use their plasmids, and what you learn in genetics is how to open up that plasmid, insert the gene you want, and then to ligase it shut, and then to put it into the E. coli. There we go, and then you get that transformation. Because you now have created inside that E. coli, by adding the human gene into the E. coli, you have created a chimera, which comes from Greek mythology, which means a creature made of many other animal parts. In this case, it's referring to the fact that the E. coli is bacterial, but it's also carrying a human gene, and that's what makes it a chimera. All right. Now, here's the problem. We might have a couple of them, and this is how or this is what I would ask. So I would say, I'm going to tell you the name and the action of a specific restriction endonuclease, such as, let's go down to here. BAMH1 recognizes, this is what will be on the exam, the DNA sequence 5 to 3' of GGATCC, and it cuts after the first G. So you're going to circle. Now, I couldn't figure out how to circle, so I put an underline. On the sequence of the DNA, that's going to be recognized by the restriction endonuclease, and then you're going to put a slash between the nucleotides cut. So here's double-stranded DNA, and this is 5' to 3', and know that the bottom strand is opposite, it runs anti-parallel, so it's 5' to 3'. So I'm looking down this, and I'm looking for GGATCC. This is the only place that I find it, so I underline it, and I know it's going to cut between the two G's, so I put a slash. The bottom strand, and I will mark these 5 to 3, 5 to 3. You got to go backwards. And so when you go backwards, you've got GGATCC, and you've got to put a slash between the two G's. That's all you have to do. All right, this just goes into humulin. I might just add 1 2-point question on that. For this could be a matching. The stages of genetic engineering has 4 stages. Know them, know what happens, and then match them up. It's going to be, what the stages are. A could be DNA cleavage, B could be production of recombinant DNA, C could be cloning, D could be screening. Okay, and the screening could have primary, E, and F could be secondary. Okay, so the matching, what is the function, what's going on? So under DNA cleavage, you cleave with a restriction endonuclease, and it's going to recognize a sequence and cut it into fragments. Stage 2, production of recombinant DNA. In this one, you're going to put that DNA fragment of foreign origin, like humulin, human insulin, inserted into the plasmid. Now, I haven't talked about viruses, so it's not going to be on the exam, just the plasmids into bacteria. Cloning, you copy the gene. In other words, you put the bacteria into broth, you grow them up. They now all clone because bacteria don't do sex. They just do cloning. They just copy themselves. And then screening has 2 steps. Know the 2 steps, primary and secondary. Now, I'm going to use what we did in lab as examples on the exam. The primary screening was the antibiotic resistance. You had plates that had ampicillin added to the media. So when you added the bacteria, if it did not have the plasmid with the gene for resistance, they died. If they had the gene for resistance, they lived and grew on the plate. And you did a fantastic job, and that's exactly what you got. So the primary screening is usually antibiotic resistance. Secondary screening is of the gene of interest. So our gene of interest was the pGlow gene, and if you successfully got that into the E. coli, it glowed in the dark under UV light. So that's the secondary screening. That brings us to the last, and the last is CRISPR. And CRISPR is a gene editing system, but originally, and it is still, it is a way that bacteria can, can have an immune system that can recognize foreign DNA like viruses that are trying to attack, destroy, take over the bacteria. So this is a very primitive immune system that the bacteria have evolved, that we now have figured out and are using it in a different way. So CRISPR-Cas9 genome editing tool, a lot of this is going to be multiple choice. It's faster, cheaper, more accurate than other types of gene editing, has enormous implications, and it enables geneticists to edit very specific parts of the genome. They can either remove, add, or alter sections of the DNA sequence. The mechanism, so CRISPR-Cas9 contains two key molecules that can produce a change in the DNA. And the first one is a nuclease, and I want you to know that Cas9 is the name of the nuclease in the CRISPR system. It acts as a molecular scissors. It can cut the two strands of DNA in a specific location so that bits of the DNA can be added or removed or altered. So let's go to the next slide. All right, the next thing I want you to know is guide RNA. Now, this RNA will bind to the sequence in question and guide it to the right spot in the DNA in the chromosome. And this then makes sure that the Cas9 enzyme cuts at the right point. In other words, the guide RNA guides the nuclease to the site on the DNA that you want to cut. Then it's going to find and bind to the sequences that you want. The RNA bases are complementary, right? A binds with U, G binds with C. And so, in theory, guide RNA will only bind to the target sequence and none of the other regions of the genome. That's a big piece that CRISPR is very good at, guiding the nuclease to a very specific gene on a very specific chromosome, and that specificity is what makes it so exciting. All right, so at this stage, the cell recognizes that the DNA is damaged and tries to repair it. And of course, what we're trying to do, not the bacteria are trying to get rid of a virus. We're trying to get rid of mutated genes that are causing diseases like Tay-Sachs, or like type 1 diabetes, or like Parkinson's disease, or like cystic fibrosis. These are damaged genes, and we have not been able to edit them prior to CRISPR. We've not been able to successfully cut them out and replace them with a therapeutic gene, but we can now. Right now we're doing it mainly in animals. China is kind of walking that ethical line and is doing it in humans, and it's going to be the miracle in your generation. So, so the DNA that is damaged tries to repair it, and scientists want to use that repair machinery to introduce changes to one or more of damaged genes, mutated genes, genes that are causing disease in the genome of interest. Now, there is one other that I want you to know, and that's called PAM, the protospacer adjacent motif. Now, do you see how it says protospacer adjacent motif, parentheses, PAM? That is what I'll do in this exam. Some of you are going to memorize Pam, other are going to recognize protospace or adjacent motif. I'll put both in the exam, like right here. This is a DNA sequence immediately following the sequence targeted by the Cas9 nuclease in the CRISPR bacteria adaptive immune system. And essentially what that says is, alert, 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 this is viral DNA, this has to be removed, alert, alert, alert. And we can also create little PAMs to see, it helps the guide RNA. So when you have this little alert, 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 then the PAM will guide the nuclease to that PAM site that says to the guide RNA that this is foreign, this is different, this has to be cut and eliminated or repaired or whatever. Now, if it's virus, believe me, they cut and eliminated. So then PAM is PAM is a component of the invading virus in the bacteria, but then we can create PAMs in order to get the nuclease to the right spot in the right chromosome on the right gene, since we're dealing with 38,000 and a bacteria only has a couple of hundred genes. So we have to be a little bit more eloquent or sophisticated in our PAMs, but we're doing it. So then, but you see, that was what says it's foreign, it's different. And when we create PAMs, we're tagging those deficient, defective genes that need to be replaced. And then the guide RNA can come whipping in, and the nuclease can cut it, and then we can apply and then we can expose it to the right sequence, and we get that into the DNA, and the person is cured of cystic fibrosis. Wouldn't that be wonderful? But that's what they're aiming for. All right, so Cas9, the nuclease, will not successfully bind if it's not followed by the PAM sequence. And then, of course, that's targeting, saying destroy, destroy, destroy. And then the DNA that we want to get rid of, we'll be able to cut as it's being guided in by the guide RNA. Complicated, but it works. So just, again, three things are going to happen. PAM, we can create them, and they will target the part of the gene in the chromosome that they want the guide RNA to bind to. Then the guide RNA bound to that PAM sequence will allow the Cas9 nuclease to cut it. Then we can bring in the therapeutic gene and join them up. Okay, applications. Again, this is just about the fact that the CRISPR came about because bacteria did not want to be destroyed by viruses. So it's the most primitive immune system in living organisms. And so that is what the bacteria do. We're going to use the gene editing system of CRISPR to apply it to humans and human diseases. And so this goes into, this is like 1.1 multiple choice question. And the applications are only going to be a couple of multiple choice questions. So anything that has a mutant gene, and this could be hypercholesteremia, hepatitis B, because we can get rid of viruses too, cancer, genetically inherited diseases like cystic fibrosis. And see, that's the other thing too, is that you can have germline cells that make egg and sperm also be treated by CRISPR. That is going to be ethically a little bit shady. Let me get into it at the last sentence here. Right about now, carrying out gene editing in germline cells, eggs and sperm, sex cells, gametes, is illegal. It's illegal in the US, it's illegal in the UK, it's illegal in most of Europe. However, all right, so however, you can do it in a living person and not a germline cell. And in a living person, a child who has cystic fibrosis, they could be treated with CRISPR and not treat their germline cells, I mean, good God, this child could be 1 year old and they have cystic fibrosis, and then they are treated especially with their lung cells, to clip out the defective gene, put in the right gene, and that child is cured of cystic fibrosis. But if they end up reproducing, then that is going to be something that they would have to be tested to make sure that they are not passing that on. Curing a person during their lifetime doesn't necessarily mean that their germ cells are altered. However, this last one. Controversial use of CRISPR came from a lab in China, this is last year, where embryos were treated with CRISPR to remove a gene that enables HIV infection. Two twin girls have been born with these modifications. So what the Chinese did is they were able to do in vitro fertilization. And then after in vitro fertilization, the fertilized egg starts to grow and divide. And at that stage, you don't have a fetus. At that stage, what you have are embryos. And that these embryos came about prior to by gene editing the egg and the sperm in the lab. So you would have a woman super ovulate, you collect the eggs, you do CRISPR. You would have a man donate his sperm, then that would be CRISPR. Then the egg and the sperm would come together, both of them modified by CRISPR. Then you would have the embryos. And then the embryos could be planted to a woman who could bring them to term. And that woman would not be related, and the egg and the sperm would give you, in this case, two girls that seemingly have had a genetic manipulation by CRISPR that enables them to not be able to be infected by the HIV virus. A couple of comments. How in God's name do you test this? Are you going to deliberately infect the person, these two girls with HIV to see? I realize that perhaps you could do it in the lab, but really? The other thing is that that's totally illegal. And the interesting thing about this is that once the world got news that the Chinese have actually used CRISPR to modify genes in eggs and sperm, and then did in vitro fertilization, and then used women as donors, not donors, women as surrogates, and actually gave birth to two girls, and they were able to confirm that these girls had this CRISPR modifications in their DNA, the world condemned them completely, and the scientists, the Chinese scientists, disappeared. And we don't know where they are, especially the head scientist. He's just poofed, and no one can find him. Oh, so on that happy note, CRISPR gene editing tool, along with epigenetics, is really going to revolutionize the way that medicine is done as you grow older. We are still obviously working out a lot of the development of drugs, how you should test it, in what people, patients, embryos should be tested. And having said all of that, that is it for that last exam. So I will see you on Wednesday. Take care. Bye-bye.